how your glucose is being utilized for energy. We started with glycolysis and the products of glycolysis, reducing equivalents, NADH goes to the respiratory chain. And you know that your pyruvate, which is the product of your glycolysis, goes to, again, also to the mitochondria to be converted to acetyl-CoA. And acetyl-CoA inside the mitochondrion is the substrate for your Krebs cycle. And in the Krebs cycle, you generate reducing equivalents, NADH and FADH2, and they also go to the respiratory chain. Okay? So that's how your glucose brings about the conversion. The fuel molecule glucose is converted now to energy and captured in a form of ATP. Okay? All right. And then we said there's plenty of glucose, and this glucose now is being stored as glycogen through the process known as glycogenesis. And then when the blood glucose goes down, especially between intervals of eating, breakfast, then you have your noon meal, and then from your noon meal to your supper, then in between, unless of course you're almost always eating, no? But usually we have only 3 full meals, breakfast, dinner, and supper. Now your dinner has been changed to lunch, no? And the dinner becomes an evening meal. I don't know why it's like that. Otherwise, if dinner is the evening meal, then you do not have last supper. You will have last dinner, no? So we know that the last supper is the evening meal. Okay. All right. So whenever there is a lowering of your blood glucose, the glycogen reserve are being utilized and broken down to glucose. And this is done in the liver. Although there is also glycogenolysis in the muscle, but the product there is not free glucose. It is glucose 6-phosphate, and it is utilized by the muscle only. While in the liver, the product of glycogenolysis is free glucose, which goes to the bloodstream to augment or to increase the concentration of glucose in the blood. So you increase the blood glucose content. Okay? All right. So that's how it is. Now, what will happen if there will be plenty of glucose? We said it is stored as glycogen. However, if there is still glucose, so where will this glucose go? Your liver is already full of glycogen, your muscle is full of glycogen, and yet there's still so much glucose. No? So we go into this pathway known as the pentose phosphate pathway, also called your Hexose Monophosphatian. Now this pathway is for oxidation of glucose, but no ATP is formed. So the glucose is just oxidized, but it is not made into ATP. However, in this pathway, there are two products that are quite important. Okay, remember, two products. It does not form ATP. Now the products here, one is NADPH. It is a phosphorylated NADP. And then this is reduced to become NADPH. This is a reducing compound. It is a reductant which is needed in the synthesis of bigger molecules out of small molecules. Okay? Like for example, fatty acids. Fatty acids are made up of acetyl-CoAs. It all depends on how many acetyl-CoAs, no, for that can be utilized for a certain amino acid, fatty acid, I'm sorry. Certain fatty acids. Like for example, you have stearic acid, which is made up of 18 carbons. No? And this require 9 acetyl-CoAs to be put together. Okay? Now, in this putting together the acetyl-CoAs in order to form a fatty acid like stearic acid, you need a reductant. And that reductant is not NADH, but rather the cousin, which is phosphorylated, which is NADPH. So there's a difference between these two. NADH is for making ATP. No? Because you know your NADH goes to your electron transport chain or your respiratory chain. But NADPH does not go there. NADPH is used for putting together small molecules like acetyl-CoA to form a bigger molecule like stearic acid or a fatty acid with 18 carbon atoms. You see? So you require that. And the other product, first is NADPH. The next product is Ribose 5-phosphate. So it is Ribose 5-phosphate. It is a pentose with a phosphate. Now what's so important about this? Ribose 5-phosphate is the sugar that is utilized in forming nucleotide. And you know what nucleotides are? Nucleotides are put together to form nucleic acid. Okay? So it becomes now a bigger molecule. Now, because your Ribose 5-phosphate will have to acquire a nitrogenous base, either a purine or a pyrimidine, and then it is phosphorylated, and a good example of that is ATP. ATP is adenine, that is for a triphosphate. Okay, but you have to have the sugar, and the sugar there is Ribose 5-phosphate. So it is adenine, Ribose 5-phosphate, and your 3 phosphates. Okay? So, what does this tell you? This tells you that the organism is growing. Okay? Di ba? Now, the organism might not, or let's say the human being may not, as soon as it attains its adulthood, it doesn't grow taller. But it can go, or it can grow sideways. Di ba? Okay, so that's fatty acids. And you need fatty acids to form your neutral fat, triacylglycerol, in order to grow sideways. Di ba? And now, if the cell is going to subdivide, you have this in your kindergarten genetics. When a cell is going to subdivide, the DNA in the nucleus of that cell has to be replicated. So meaning to say, you will have to create a new DNA, a copy, an exact copy of the original DNA. And you need nucleotides. And where do we get nucleotides? First, you need to have ribose 5-phosphate, and then a purine or a pyrimidine base, and then several phosphates. And you have the nucleotide. The nucleotides are put together to form your nucleic acid DNA. So now, if there are already two DNA in a cell, the cell now subdivides. And when it subdivides, the two DNAs are separated. Each daughter cell will have its own DNA. So that is growth. Diba? All right, so that is what is in this chapter. Your pentose phosphate pathway and we will also talk about other pathways of Hexose metabolism like galactose, fructose. Okay. Importance. Okay, so I have mentioned it already. NADPH, synthesis of fatty acids, and steroids. And in the steroid, you include cholesterol. No? Okay, cholesterol. So, cholesterol is made up of acetyl-CoAs together. So if you have a high cholesterol, what's the first thing that you should do? As much as possible, you should not have so much of acetyl-CoA. And the acetyl-CoA that is used is the acetyl-CoA from carbohydrates or from glucose. You can also get acetyl-CoA from the breakdown of fatty acids. So you'll have plenty of acetyl-CoA. But this breakdown of fatty acids, acetyl-CoA, cannot be put together to form cholesterol or fatty acids. Why? Because when you utilize your fatty acids as a fuel, it means you are fasting or there is no food. So you have to utilize your store of fuel, and that is your fatty acids. But where is the glucose? The glucose is used primarily in fasting state for the maintenance of energy needs of your red blood cell, because your red blood cell cannot use any other fuel except glucose. Now, The nervous tissue, yes, they use glucose, prefer glucose, but when the glucose content of the blood is very, very low, your nervous tissue, including your brain, shifts to the use of ketone bodies. The idea there is to preserve whatever glucose that is left for maintenance needs of energy for the RBC. You get me? Now going back to cholesterol. Cholesterol is made up also of acetyl-CoA. But it requires NADPH. And where do you get NADPH? It's from glucose. Diba? Now you say acetyl-CoA from fatty acids, it cannot be because you cannot break down your fats unless there is no more glucose in the blood. When the glucose content is very low, that's the time your body uses fats. And when there is no more glucose, for pentose phosphate pathway to produce NADPH, you cannot put together that cholesterol, no, to form cholesterol from acetyl-CoA because you do not have NADPH. Did you get it? Huh? So high cholesterol, minimize your carbohydrate intake. That's number one. All right. And the other product is ribose. Okay, so you can see it here. Ribose for nucleotide and nucleic acid formation. Glucose, fructose, galactose are the main hexoses absorbed from the gastrointestinal tract, derived from dietary starch, sucrose, and lactose. Okay. Fructose can be converted to glucose. Isomerization, it's just an isomer, glucose. Galactose is also an isomer of glucose. So an isomerase can convert galactose to glucose. Okay? There's another one here, something that's very important also, because we're talking about biomedical importance. It says genetic deficiency of glucose 6-phosphate dehydrogenase, the first enzyme of the pentose phosphate pathway, causes acute hemolysis of the blood cells, resulting in hemolytic anemia. How can that be? Okay, let's just talk about this one here. So again, sir, this is an alternative pathway for oxidation of glucose, does not lead to ATP. Two major functions: number one, NADPH; number two, ribose. Okay. This glucose 6-phosphate dehydrogenase, the first enzyme of your pentose phosphate pathway, produces right away NADPH. We know already how important is NADPH. But there is another very important. You know, your red blood cell carries oxygen. Okay? And oxygen can bring about a formation of free radicals from oxygen. That's the superoxide. No? And if you have superoxides, it breaks. It's going to break what you call the membrane of the RBC. And when the RBC, the membrane of the RBC breaks, and of course, that is what we call hemolysis. And as a result, your RBC are hemolyzed, leading to what we call hemolytic anemia. Diba? Now, where does NADPH come in here? The integrity of your membrane of your RBC is guarded by your glutathione, guarded from the onslaught of the superoxides or other free radicals in which the RBC are subjected to. So, that is oxidized, and that oxidation of your membrane brings about hemolysis. Okay? Now, it so happened that there is an antioxidant. And you know that antioxidant is very, very popular with the ladies because they use it, no? And that is glutathione. That is the important function of your glutathione, to preserve the integrity of the cellular membrane. That is how important glutathione is, the cellular membrane integrity. So once the cellular membrane of the RBC is broken, naturally hemolysis comes around. All right? Now, in doing so, your glutathione becomes oxidized. And when it becomes oxidized, after its encounter with the free radicals, when it encounters the free radicals, it becomes oxidized. So, that glutathione is useless unless it is reduced by NADPH. Okay? NADPH. So the NADPH here is quite important in restoring the oxidized glutathione to become reduced glutathione so that it can continue its work in preserving the integrity of your cellular membrane of the RBC. You see? Now, another importance in the uronic acid pathway. Now, this has not got to do with your pentose phosphate anymore. This is another pathway of glucose, no, to be oxidized, but no ATP is formed because there's plenty of glucose already. But this pathway will not come into action unless there's plenty of glucose. So, uronic acid pathway. So, this forms a very important conjugating compound known as glucuronic acid. You remember glucuronic acid when we were talking about xenobiotics, which is a conjugant, which conjugates non-water soluble compounds to make it soluble in water. So you have glucuronic acid, and we call them glucuronides. Okay? It's xenobiotics, still remember. Excretion of metabolites and foreign chemicals. Xenobiotics. No? A deficiency in the pathway leads to condition of essential pentosuria, the lack of one enzyme. Now, this uronic acid pathway is a pathway in lower animals, which they can produce vitamin C. Vitamin C. But in humans, the absence of this enzyme in the uronic acid pathway, this enzyme here, gulonolactone oxidase, in primates, that includes monkeys, no, apes, gorilla, and us. We cannot form ascorbic acid. That is why ascorbic acid becomes a vitamin. So it is a dietary requirement. No? All right. Also, there are minor genetic disorders like some enzymes in the fructose and galactose metabolism can lead into essential fructosuria, hereditary fructose intolerance, and galactosemia. Now, of course, they are genetic disorders because enzymes are insufficient or absent. Okay? All right, now let's go to the pentose phosphate pathway. All right, this is pentose phosphate pathway forms NADPH and ribose phosphate. Oh, here you are. The flow chart of the pentose phosphate pathway and its connections with the pathway of glycolysis. So this is connected to glycolysis. Now look at this. This is what I just want to emphasize here. Glucose 6-phosphate dehydrogenase. Now in pentose phosphate pathway, 3 glucose molecules are right away taken up. And you know that dehydrogenase, it is oxidized, NADPH is formed, NADPH is formed, NADPH is formed right away. And not only there, the second enzyme that acts on it also produce NADPH, 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 and at the same time removes 1 carbon in the form of carbon dioxide. So from a 6 carbon, a hexose, now from here it becomes a pentose. Okay, so what are the pentoses you have here? Now this one here, 3 epimerase produces cellulose 6-phosphate, cellulose 5-phosphate, but this one, ketoisomerase, produce ribose 5-phosphate. And where will this ribose 5-phosphate? Some of it goes to the synthesis of RNA and DNA. Did you see that? And some portions unite with cellulose 5-phosphate and ribose 5-phosphate to produce glyceraldehyde 3-phosphate and pseudohyptulose. Pseudohyptulose. Okay? Pseudoheptulose is 7 carbon. So if you still remember our sugars, we divide the monosaccharides into whether they have 3 carbons, 4 carbons, 5 carbons, 6 carbons, and 7 carbons. Now, so this is only found in nature from 3 to 7. The rest are synthetic. So if you have a monosaccharide which has 8 carbons, that is synthetic. It is not natural. Synthetic means it is made in the laboratory. Addition of 1 carbon at a time. They can even go 9 or 10 carbons or 11 carbons or 12 carbons. Those are all synthetic. Okay? So, natural carbons for the monosaccharides are only from 3, 4, 5, 6, and 7. So this is your pseudoheptulose phosphate. Okay? Now, if you look at this, there is a restoration. There is a restoration of the intermediates into glucose. Glucose 6-phosphate, glucose 6-phosphate, and 1/2 glucose 6-phosphate. So what is 1/2? It's a triose. So glyceraldehyde triphosphate or trihydroxyacetone. No? Okay? 1/2. Okay. So this 2 glucose 6-phosphate goes back again. And then we know that it started with glucose 6-phosphate, 3 glucose 6-phosphate are utilized right away. Okay? The pathway is indicated consists of 3 interconnected cycles with glucose 6-phosphate as both substrate and end product. The reactions above, the Brooklyn error non-reversible, whereas all reactions under that line are freely reversible apart from the catalyzed fructose 1,6 biphosphatase. Okay. So what is important is the first 2 reactions produces NADPH, 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 NADPH. And of course, the formation of ribose 5-phosphate for synthesis of RNA and DNA. As the importance of your ribose 5-phosphate. So the reactions of the pentose phosphate pathway occur in the cytoplasm. What other pathway is in the cytoplasm that belongs to glucose metabolism? Glycolysis. So there is interrelation with the pentose phosphate pathway and glycolysis. Okay, like glycolysis, the enzyme, the pentose phosphate pathway are found in the cytoplasm. Oxidation is achieved by dehydrogenation using NADP, and it is emphasized not NAD as the hydrogen acceptor. Sequence of reactions in the pathway may be divided into phases. Irreversible oxidative phase, those are the first two, and the reversible non-oxidative phase. The first phase, okay, glucose 6-phosphate undergoes dehydrogenation and decarboxylation. In the second phase, ribulose 5-phosphate is converted to glucose 6-phosphate. You see that? Reactions involving mainly two enzymes, transketolase and transaldolase. So the oxidative phase of your pentose phosphate pathway generates NADPH. Okay. So this is the one that is your, where you'll find the carbons, no? Carbons are how it is born. Now, I want you also to note that the pentose phosphate pathway is also dependent on thiamine. Thiamine. What is thiamine? What vitamin is thiamine? Vitamin B1. Vitamin B1 is part of a metabolism of carbohydrate. Vitamin B6 for amino acids. No? And there are other vitamins later on. Okay? Non-oxidative phase. Okay, now this is a problem. Although glucose 6-phosphate is common to both pathways, the pentose phosphate pathway is markedly different from glycolysis. Oxidation is NADPH, while NADH is glycolysis. No? And carbon dioxide, which is not producing glycolysis. No ATP is generated in your pentose phosphate pathway, whereas in glycolysis, ATP is formed. Substrate level phosphorylation. Two pathways are however connected. Cellulose 5-phosphate activates protein phosphatase. Okay, this goes on. All right. Let's leave that alone. Never mind that. Reducing equivalents are generated in those tissues specializing in a reductive synthesis. What do you mean by this? Synthesis, you know what is synthesis? Making bigger molecule out of small molecule. But when you say reductive, so it needs a reductant. And the particular reductant is in NADPH. Okay? So, where is pentose phosphate pathway active? Particularly in the liver. There are so many synthetic reactions in the liver. Adipose tissue, fatty acids. Adrenal cortex, the steroids. Thyroid, your hormone there. Erythrocytes. Erythrocytes for your, we have mentioned that. It's about hemolytic anemia. The testes. Oh. And the lactating mammary gland. Lactating mammary gland. Its activity is low in non-lactating mammary gland. So it is active mammary gland. It is low in non-lactating mammary gland and skeletal muscle. These tissues in which the pathway is active uses NADPH and reductive synthesis. Fatty acid, steroids, amino acids, and reduced glutathione. Okay? The synthesis of glucose 6-phosphate dehydrogenase and 6-phosphogluconate dehydrogenase may also be induced by insulin. So, what is this tell you? When is insulin secreted? When you have high glucose in the blood. And that is, of course, which induces glucose 6-phosphate dehydrogenase for your pentose phosphate pathway, and also this enzyme, 6-phosphogluconate dehydrogenase, which is in the feed state and lipogenesis increases. Ribose can be synthesized in virtually all tissues, of course. No? That is, of course, for nucleotides, because it is not only nucleotides that are being used for forming nucleic acids. What about ATP? What about GTP? Those are nucleotides. No? And you know that your ATP is, of course, your currency for energy. Okay. So all cells, almost all cells, can synthesize ribose through pentose phosphate pathway. Okay. Pentose phosphate pathway and glutathione peroxidase protect. Okay. We have this, I mentioned this already. Protect erythrocytes against hemolysis. The pentose phosphate is the sole source of NADPH for reduction of oxidized glutathione. And this is catalyzed by glutathione reductase. Okay. So when the glutathione is oxidized because it tries to blunt the effect of free radicals, they become oxidized. So in order that they still can be useful, they have to be reduced. And you have an enzyme, glutathione reductase, which uses NADPH as the reductant. Selenocysteine here is also very important. Reduced glutathione removes, reduced hydrogen peroxide, a reaction catalyzed by glutathione peroxidase, an enzyme that contains the selenium analog of cysteine, selenocysteine at the active site. The reaction is important since hydrogen peroxide accumulation decreases the lifespan of erythrocyte by causing oxidative damage to the cell membrane, leading to hemolysis. Okay? And because of the presence of your glutathione, you reduced, the glutathione there reacting with the free radical hydrogen peroxide becomes oxidized. And so it has to be reduced by your glutathione reductase, which requires NADPH. Oh, so this is the reaction here. See, pentose phosphate pathway produces NADPH, and the enzyme reductase. Oh, well, now up to, this is your glutathione, and this is your oxidized glutathione. And you need a glutathione peroxidase here. If you look at this hydrogen peroxide, and then you form reduced glutathione, and that reduced glutathione becomes, oxidized glutathione becomes reduced by glutathione reductase, and you have now reduced glutathione and acts on your hydrogen peroxide again through the enzyme glutathione peroxidase. So you can just see how important is this one for formation of NADPH and resulting in prevention of the breakdown of the cell membrane of erythrocytes. Otherwise, hemolytic anemia will ensue. So here again is pentose phosphate pathway in a nutshell. So repeat it again, NADPH for reductive biosynthesis, protection against oxidative damage, and Ribose 5-phosphate for nucleotide production. Like glycolysis, it is present in every tissue. Okay, in the irreversible oxidative phase of your pentose phosphate pathway, 1 carbon of glucose 6-phosphate is released as carbon dioxide, NADPH is generated, and ribulose 5-phosphate is produced. Ribulose 5-phosphate provides Ribose 5-phosphate. So your ribulose 5-phosphate becomes Ribose 5-phosphate for nucleotide biosynthesis. Or generates pentose phosphates, the other phosphates, cellulose phosphates, which enter the non-oxidative portion of the pathway. In the non-oxidative reactions, it produces ribulose 5-phosphate may be converted to intermediates of glycolysis. Ribulose 5-phosphate may be isomerized to form Ribose 5-phosphate or epimerized as an epimer to cellulose 5-phosphate. Okay? So the oxidative reactions are irreversible. However, the non-oxidative reactions are reversible and therefore can be used to generate ribose 5-phosphate for nucleotide synthesis from intermediates of glycolysis. Okay? There is a connection between glycolysis and pentose phosphate pathway. The reactions of the pentose phosphate pathway, oxidative reactions, the first two enzymes. No? You just look at those, the pathway there, and you can see the enzymes involved. And products of your oxidative reactions, carbon dioxide, and NADPH. Okay, so this is the non-oxidative reactions. As we said, they could be converted to intermediates for your glycolysis. That's why there is relationship between pentose phosphate pathway and your glycolysis. Here's the summary of reactions. This is the overall reaction. 3-glucose 6-phosphate plus 6-NADP. Ribose 5-phosphate plus 3-carbon dioxide plus 6-NADPH. Ribose 5-phosphate for nucleotide synthesis. So here we are, NADPH for fatty acid synthesis. NADPH is also used to reduce glutathione. When NADPH are low, the oxidative reaction of the pathway may be used to generate ribose 5-phosphate for nucleotide biosynthesis. Reversible non-oxidative portion of the pathway can be used to generate ribose 5-phosphate for nucleotide biosynthesis from fructose 6-phosphate and glyceraldehyde 3-phosphate when NADPH levels are high. Okay, so we are now in uronic acid pathway. Glucuronate, a precursor of proteoglycans and conjugated glucuronides, was the product of uronic acid pathway. So uronic acid pathway, importance there is glucuronic acid and ascorbic acid, except in us and other species for which ascorbate is a vitamin. It is an alternative oxidative pathway for glucose. Now, like pentose phosphate pathway does not lead to the formation of ATP. So glucose 6-phosphate is isomerized to glucose 1-phosphate and so on. No? So we just follow the pathway. There you are. Okay. So this is the product, glucuronic acid, used as a conjugate. Now, it is reduced and becomes gulonate. It is dehydrated, becomes gulonolactone. But the oxidase that is to be the enzyme here is absent in primates. It is absent. You know what are primates? That includes us. Now, a dog generates 20 grams of vitamin C daily. Can you imagine that? Vitamin C, ascorbic acid, 20 grams. That would mean 20,000 milligrams. Oh, that's why dogs are very hardy. Not, of course, your dog that are given dog food and also those, but the ascals. The ascal, they just go to the garbage dump and then they eat anything there. And they do not get sick. Yeah? Okay, so there you are. Now, there is a, there's something here in the vitamin C. A lot would say that to not increase your vitamin C consumption because it might lead to oxalate formation, and oxalate with sodium becomes sodium oxalate or other metals, and it becomes stone, oxalate stones. No? As you can see it here. From ascorbic acid, here ascorbic acid in the dogs, then oxalates are formed. Okay? But have you seen kidney stones in the dog? 20,000 milligrams a day. And yet there is no oxalate, calcium oxalate stones. No? So it's here. If you form this dihydroxyascorbic acid, and decarboxylation, carbon dioxide, this is black in humans, so result into formation of oxalate. Now, in the study of vitamin C, one famous researcher, Linus Pauling. Oh, what did Linus Pauling did? What was he doing? He was researching on vitamin C. Okay, and he was taking vitamin C and looking at his urine for calcium oxalates, and he never found any calcium oxalates in his urine. He has reached 10 grams That's 10,000 milligrams. And I never had any calcium oxalate stones. Now, because of that work, Linus Pauling had what you call this, received the tsaka itong award in medicine for research. I forgot the award. So he got that award 2 times in a row for vitamin C research. And what was he doing? He was using himself as a guinea pig. Okay. So that's the importance there, no? Of uronic acid pathway, glucuronic acid, and the enzyme that is blocked, gulonolactone oxidase, so that we cannot form ascorbic acid. Okay? All right. Ingestion of large quantities of fructose has profound metabolic consequences. Have you taken in fructose syrup? High fructose syrups used in manufactured foods and beverages. Large amount of fructose entering the hepatic portal vein. Why is this fructose and then enclosed in parentheses glucose? Any idea? Yeah, look at that, fructose, high fructose. And why is this enclosed in parentheses glucose? Well, yes, that's fruits, no? But what we have here is high fructose syrup coming from corn. High fructose syrup. And this is the one now that is used as a sweetening agent. Because, you know, fructose is very, very sweet. And it is very, very cheap. They call this magic sugar. It's magic sugar because a little amount only is being used. And it becomes very, very sweet. It's fructose. And that is why we are concerned with COVID. But what is disturbing is we have an epidemic of diabetes. It's worldwide. You know, fructose and glucose are isomers and they can be interchangeable. It can interchange right away. Your cells are capable of converting fructose into glucose. Even in the absorption, even in the intestinal wall, when fructose is absorbed, it is converted right away into glucose. Much more, if there are still fructose that goes into the liver, that fructose is converted to glucose. Okay? Although fructose undergoes more rapid glycolysis in the liver than does glucose because it bypasses the enzyme phosphofructokinase. This allows fructose to flood the pathway in the liver, leading to increased fatty acid synthesis. No? Why? Because there's plenty of acetyl-CoA that will be formed. Because glycolysis is very active, produces pyruvate. Pyruvate goes into the mitochondrion. In the mitochondrion, it becomes acetyl-CoA. And acetyl-CoA, because there's plenty of glucose, NADPH is formed, and NADPH is used to put together your acetyl-CoAs. And now you have fatty acids. Okay? And then these fatty acids is esterified with your alcohol. And what is that alcohol? Is glycerol, which you manufacture. And you form it now triacyl glycerol, in which it ultimately increases LDL cholesterol concentration. Okay, fructokinase in the liver does not act on glucose, but only on fructose. Okay, so in the liver, kidney, intestine catalyzes phosphorylation of fructose to fructose 1-phosphate. This enzyme does not act on glucose. See? Unlike glucokinase, its activity is not affected by fasting or by insulin. You see that? Which may explain why fructose is cleared from the blood of diabetic patients at a normal rate. Then fructose is cleaved into glyceraldehyde and dihydroxyacetone by aldolase B. This is glycolysis. Enzyme found in the liver, which also functions in glycolysis in the liver. And so on. The 2-triose phosphate, dihydroxystone phosphate, glyceraldehyde phosphate may either be degraded by glycolysis or may be substrate for aldolase, and hence gluconeogenesis, which increases, of course, the glucose content. So that's the problem with high fructose syrups. And this can be sourced very cheaply. That is the reason why our sugarcane industry is no longer very lucrative, because there is now a substitute, high fructose syrup from corn. The corn industry in the United States or in other countries is not used as a staple food. Unlike here in the Philippines, where we have utilized corn, no? Because it is cheaper than rice. Okay? But the corn there, they grow corn instead of sugar cane, because the corn can be converted to high fructose syrups. Very, very cheap. So our sugarcane industry now has a problem because you have a competition. Sugarcane is no longer lucrative because they found a way how to produce fructose from sugar cane. Or I mean from corn. And this is very, very cheap. And this is now used in the manufacture of foods to sweeten the food and beverages. Beverages. So your soda drinks is no longer from cane sugar, but from 5 fructose syrups. And as a result, we have an epidemic of diabetes mellitus, including here in the Philippines. Especially Filipinos has a sweet taste. Diba? Our humba does not taste well if it is not with sugar. Okay, so if you look at this is glycolysis. Remember? Now glycogen, glucose 6-phosphate, and then fructose 6-phosphate, and so on. Split into two, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, and then it becomes pyruvate. Now here, look at this. It becomes fatty acid synthesis. This is a shortcut because that pyruvate has to become acetyl-CoA first. And it has to be done only in the mitochondrion because the pyruvate dehydrogenase complex is found only in the mitochondrion. And that's how you get your acetyl-CoA. See? Okay. So here is your fructose from the diet, from the soda drinks that you are drinking. Okay, fructose goes directly in here. It bypasses this one. So it goes down here. That's the dotted line only. Fructose acted by hexokinase becomes fructose 6-phosphate and goes through your glycolytic pathway. But here, it goes this way, and then that way. You see? Oh, this is a shortcut. And then acetyl-CoA fatty acid synthesis. All right, of course, when you do not have this enzyme here, and this one, the red one, then you have the consequence of fructosuria. No? Now this one is black and hereditary fructose intolerance. That is fructosuria. This one, fructose intolerance. It is just like lactose intolerance. But again, genetic disorders are rare in Asians. In Asians, it's rare. Unlike in Europeans, they have plenty of this. And you know the reason why. Now, why they have so much of genetic malfunctions? Because of intermarriages within the family. Okay, so that is your fructose. So again, avoid high fructose syrups, corn syrups. Galactose. Galactose is needed for the synthesis of lactose, glycolipids, proteoglycans, and glycoproteins. And it says here, what is our source of galactose? Then you will say, lactose. Now, supposing a person does not have lactose, does not take in lactose, so you cannot have galactose. Wrong. Because galactose and glucose are isomers and they are epimers as well. They only differ in the arrangement of the atoms in carbon 2, no, carbon 4, galactose and glucose. Huh? So they can be interchangeable, easily formed. That's why a lactating woman does not need to have to take galactose because the glucose can be converted to galactose. So galactose plus glucose combine together to form lactose. It's a beta 1,4 linkage for glucose and galactose. Okay. So, but, a good source of galactose is lactose, of course. And we get this from milk, mammalian milk. Okay. Disaccharide lactose, sugar found in milk. It is readily converted in the liver to glucose. Then it has a specific enzyme for phosphorylation of galactose. We have galactokinase to produce glucose 1-phosphate, reacts with UDP glucose to form uridine diphosphate galactose, UDP, and glucose 1-phosphate. Reaction catalyze galactose 1-phosphate uridyl transferase. Okay. The conversion of UDP galactose to UDP glucose is catalyzed by an epimerase, 4 epimerase. That means the 4th carbon atom. The reaction involves oxidation then reduction at carbon 4 with NAD as the coenzyme. UDP glucose is then incorporated into glycogen to form glycogen through glycogenesis. Oh, here you are. So you have a specific enzyme, galactokinase, acting on galactose. So galactose 1-phosphate, all right? Reacts with UDP galactose, UDP glucose, if we have UDP galactose and glucose 1-phosphate. So they interact with each other. The glucose here becomes glucose 1-phosphate. The galactose 1-phosphate here becomes UDP galactose. Okay? And then you have your, it can be converted by an epimerase back to UDP glucose, and then glycogen synthase forms glycogen. So this is just the intake conversion of glucose and galactose. In the other pathway, this is where lactose is formed in the mammary gland. Glucose becomes 6-phosphate through your hexokinase, which is found in the mammary gland. And glucose 6-phosphate becomes glucose 1-phosphate through a mutase, and then you have now uridine triphosphate comes in, and you have the UDP glucose, and that UDP glucose can be converted through an epimerase, and that UDP glucose can become UDP galactose, and a molecule of glucose combining the two together through a lactose synthase. And you have now lactose. Now this occurs inside the active mammary gland. So without even galactose, no, you can form lactose. Okay? Active mammary gland. Glucose is the precursor of amino sugars. Okay. There are sugars in which an amino acid is added to it. So amino sugars are components of glycoproteins, glycosphingolipids, like gangliosides, and glycosaminoglycans. The major amino acid are hexosamines, glucosamines, galactosamine, monosamine. These are the hexosamines. 6 carbon atoms, 6 carbon atoms, 6 carbon atoms. Aminated sugars, aminated glucose, aminated galactose, aminated mannose. And the 9 carbon atom sialic acid. And the principal sialic acid found in the humans is N-acetylnuraminic acid. N-acetylnuraminic acid. Okay. Oh, there you are. This is your interrelationships in metabolism of amino sugars. Okay. Form your amino sugars. Your glucosamine, then you have your converted to heparin. All right. Impairment of the pentose phosphate pathway leads to erythrocyte hemolysis. We have mentioned this already, no? Some 400 million people carry a mutated gene for glucose 6-phosphate dehydrogenase, making it the most common genetic defect, but most are asymptomatic. In some populations, glucose 6-phosphate deficiency is common enough for it to be regarded as a genetic polymorphism. The distribution of mutant genes parallel that of malaria, suggesting that being heterozygous confers resistance against malaria. So heterozygous, this what you call the having this disorder are asymptomatic. No? The defect is manifested as red cell hemolysis or hemolytic anemia when individuals are subjected to oxidative stress from susceptible levels subjected to oxidative stress, such as anti-malarial primaquine and sulfonamides. During World War II, there was, an increase of hemolytic anemia. Now, because they were given antimalarial drugs and sulfonamides, this is the sulfonamides like sulfatiazole, sulfadiazine, sulfamethasone, sulfamiracil. So the sulfa drugs. No? And when they have eaten fava beans, and we do not have this. Because of this, when they eat fava beans, they have hemolytic anemia, and the disease is known as favism. Favism. Any different mutations are known on the gene glucose exposure, leading to two main variants of favism. Afro-Caribbean variant. The enzyme is unstable so that while average red cell activities are low, it is only the older erythrocytes that are affected. So, hemolytic crisis to be self-limiting. While the Mediterranean variant, the enzyme is stable but has low activity in all erythrocytes. Hemolytic crisis in these people are severe and can be fatal. Glutathione peroxidase is dependent on the supply of NADPH, meaning pentose phosphate pathway, which in erythrocytes can only be found formed in your pentose phosphate pathway. Okay? Disruption of uronic acid is caused by enzyme defects and some drugs. Okay. So you know that you have your glucuronic acid, which is quite important. So you do not have a conjugate, a very good conjugate for removing toxic materials that are not soluble in water. So you have uronic acid, and usually we have drugs, which are, medicines which have to be excreted by using glucuronic acid. Barbiturates given to rats result significant increase of glucose to glucuronic acid. Gluconate and ascorbate, aminopyrin, antipyrin increases excretion of cellulose. Okay? Loading of the liver with fructose. Again, here we go again. Potentiate the increase of formation of triacyl glycerol, and you have a condition known as hypertriacylglycerolemia. It also increases the formation of cholesterol, and so you have a condition of hypercholesterolemia and hyperuricemia, uric acid. No? High fructose syrup. Okay. Then you will say, what about fruits? We are told to eat fruits. Yes, no problem with that. But the amount of fructose compared to just 1 teaspoon of your high fructose syrup, no? 1 teaspoon, you need about perhaps 4 to 5 apples. Can you eat 5 apples in one sitting? Dili, no? So, avoid high fructose syrup. Now, this hypertriglyceridemia, glycerolemia, hypercholesterolemia, hyperuricemia, you find this in diabetes mellitus. As the reason why there is an increase of diabetes mellitus. And they even say that we already have an epidemic because this is worldwide. And together with that, imagine with so much fructose, which can be easily converted to glucose. No? So you will have a blood sugar very, very high. Just imagine that this sugar goes inside your RBC. No? And what happens is that you have you have your carrier of oxygen. What is the carrier of oxygen?
Hemoglobin.
Oh, no. Can you imagine if your hemoglobin is decorated with your sugars? And that is the first indication that you have high sugar in the blood when your hemoglobin is decorated with glucose. Now you will say, oh, it's not fructose, but you should remember that fructose can easily become glucose. Huh? So there you are. That's one of our problems now because of these high fructose syrups. Okay? Oh, napagyan niya ini. Fructose and sorbitol in the lens are associated with diabetic cataract. You know, sorbitol, which is the sugar alcohol of glucose and fructose. Sorbitol is the sugar alcohol of glucose and fructose. So this sorbitol can be easily formed when you have a high glucose content of the blood, because your lens contains a reductase which acts on glucose to form sorbitol. And sorbitol becomes anidose. You know what's anidose, no? A nest. Okay? For cataract formation. So it's called diabetic cataract because when you're diabetic, your sugar is high. You're the sugar central. Diabetic cataract. And glucose can become sorbitol very easily. Okay? So that is how you have your diabetic cataract. All right. Enzyme deficiencies, we know this already, you know. So inability to metabolize galactose is galactosemia. Now, so the thing here is just to avoid galactose. And how can you avoid galactose? The main source of galactose is milk. So you don't drink milk. Now, this galactose also, if it is not utilized, if you have lost or insufficient enzymes for metabolizing galactose, it can be reduced into an alcohol. And the name for that is galactitol. For glucose, the sugar alcohol of glucose is sorbitol. For galactose is galactitol. Because it also have your aldose reductase, which reduces your galactose into galactitol. And it's the same thing with sorbitol. It can also be as a nest or a nidus for cataract formation in the lens. So you will also have cataract, but this time it is not sorbitol but galactitol. Because the aldose reductase will act on glucose and galactose. For glucose becomes sorbitol, for galactose becomes galactitol. Okay? All right, so we have now the summary that is found in your text. So it is always emphasized here, no ATP is formed. Now, pentose phosphate pathway, no ATP formed. uronic acid pathway, no ATP is formed. Utilization of glucose or oxidation of glucose without formation of ATP. This only occurs when there is plenty of glucose. So the culprit here is extra rice. Okay, so we have here this summary. So this is your summary. You can read this. So we have finished our